thank you very much for the opportunity to present my group's work. I know this is a fairly informal discussion, but I hope that I do justice to the work that the students have done. So, if you did a follow-up study during the summer last year, and that captured a larger number of sampling, show a little bit of extra work beyond the paper. And if you don't like what I have to say, well, that's totally on me. So we'll have time in the discussion to go over that. Sorry. So I'm interested in micro and nanoplastics, and probably you've all heard about this from your media sources. Microplastics are small particles of plastic less than 5 millimeters in diameter. Plastics are less than 1 micron, although if you're in atmospheric chemistry, there's might be a little bit of controversy about the exact definitions of what nanoparticles are. For the purpose of this talk, we're talking about 1 micron particles. The motivation from our work comes from this picture here. So this is a figure that I've adapted from Driss and co-workers, and they were one of the first groups to look at the microfibers and polymers that are floating around in indoor dust. So, one of the few days in St. John's here is sunny. We can see little dust particles in the window, and we are obviously all inhaling this stuff. And the question is, how much of it are we inhaling, and whether there's potentially an adverse health effect. We know from work that's been done over the years on particulate matter in the environment that inhalation of particulate matter can cause major health issues like cancer and asthma. And so the question is, how much is plastics contributing to this problem? Now, as an analytical chemist, I'm always looking for things to do, right? And so there's a knowledge gap that we're specifically trying to address, and that is that the smaller particle sizes, less than 10 micron in size, they're typically not reported in the work that's been done so far on microplastics. And that's because most of the time those studies are using spectroscopic techniques like FTIR and Raman that are, to my understanding, it's somewhat challenging to measure small particles. It's important to capture these small particles because once you're below 10 micron, they're more likely to float around and stay suspended in the air, so you're more likely to inhale. There's also the possibility that once you've inhaled these particles, the smaller ones are more likely to translocate from the lungs into the bloodstream and to other organs. But I mean, that all has to be studied. So what I'm going to talk about today is our approach to filling this knowledge gap, and we'll speculate a little bit of whether this is a. So I'll jump right into the experimental. We're using accelerated solvent extraction to remove the polymer material that we're collecting on glass fiber filters that has collected particles that have been drawn through with a vacuum pump. This is a picture of our collection device. This is a cascade impactor. So you deploy this vacuum pump connected to this cascade impactor. There's 11 stages. At each stage, there's a different glass fiber filter. Particles of different sizes are separated on the base of their aerodynamic cut point. So we're able to actually get, a filter with approximately 10 micron particles going down all the way to about 15 nanometers or so. So we extract using accelerated solvent extraction, concentrate under nitrogen evaporation, and then inject by pyrolysis. So we are using experiments to measure the drift time of the pyrolysis products, and we have accurate mass measurements from the time of flight. This is a picture of the pyrolyzer mounted on top of the gas chromatograph. So this is the Gerstel's offering, and you have relatively small probes. These are, I'll show an expanded view here. This quartz pyrolysis tube can hold about 40 or 50 microliters of extract. So it is possible to roll a small amount of filter paper and directly insert it into the probe tube. You just have to have really talented students like mine to do it. So what is the benefit of doing multidimensional separation? The left side of the slide here shows a retention time, drift time contour plot. So we have our gas chromatographic retention time and our drift time through the 1 meter ion mobility cell. And we're able to nicely pick out series of pyrolysis products generated from common plastics. And we focused on 4 in this study: polypropylene, polyethylene, polystyrene, and polymethyl methacrylate. The N values just refer to the number of carbon atoms in each of those products. And you can sort of get the picture that we get a nice distribution in the gas chromatogram. We also see this nice separation in the second dimension. That's useful in separating, I think, the pyrolysis products generated from polypropylene, polyethylene, which are isomeric. So the branched compounds tend to elute a bit earlier and also have a shorter drift time. I mean, this is not a table of data that you're going to have to put to memory or anything like that. But one of the useful aspects of using ion mobility is that we can characterize the product ions on the basis of their collision cross section. So this is approximates the shape of the being detected. And so we're using this as a confirmatory metric to ensure that what we measure in environmental media is indeed a pyrolysis product from these plastics. Because we want to be sure that what we're measuring is indeed plastic and not some from another compound or breakdown of material. And so we got with reasonably decent agreement between CCS value in the sample versus in our analytical standards. This is just a summary of the method performance of the method. We have admittedly relatively wide error bars for some of these polymers. And I can tell you something that I think most people won't tell you in their papers. It's quite difficult to generate good quality calibration curves with polymers, or maybe this is obvious to the folks in the polymer group. It was quite a surprise initially. We found that in the paper, dissolving the polymers in heated toluene was effective. But more recently, we found that suspensions of particles in a methanol DCM mixture proved to be easier to work with. So just as a practical piece of advice. Our recoveries were reasonable. So the experiments were done as follows. The student would spike a known amount of the polymer onto our glass fiber filters, extract, solvent extraction, concentrate, and then inject in the instrument. And so Maheen was getting between 60 and 114% recoveries, although you can see that there's fairly wide error bars on those, just as a note. So what are we seeing? This set of figures here summarizes the size resolved results obtained from 2 indoor environments. So the first place we sampled was our lab. We just wanted to know what kind of breathing in our lab, but also, you know, as we move on to more difficult matrices like biological samples that lab air could potentially contaminate, we want to know what that signature looks like. And then we've also did a private residence, which, yeah, okay, that's my house. So there's a fair bit of plastic in both locations. We see that I would say broadly we're seeing similar quantities, within an order of magnitude across all aerodynamic cut points, and this translates to an exponential increase. We assume that the average particle size of each of these components corresponds to aerodynamic cut point, that we see this exponential increase, and that's a log scale on the right of the number of particles. So take-home message is the mass concentration is more or less constant across the particle sizes, and exponentially increases in terms of the particle concentration as you go to smaller aerodynamic cut points. And that's more or less what's been seen in other studies of microplastics in other media. So in the marine environment, for example, we see that the same kind of trend, where as you go to smaller particle sizes, the numbers increase. For human health, that is something important to think about, because, you know, number of particles increasing as with decreasing size, that means you're breathing in more of these particles, and if an individual particle can cause an effect, that could, you know, you're increasing your probability of risk. But I'll talk a little bit more about that in a slide or two. The concentrations, so if we look at the mass concentrations, we can break this up into size ranges more recognizable by atmospheric chemists or people doing environmental health applications. PM10 represents particulate matter that has aerodynamic diameters smaller than 10 micron. PM2.5 is a 2.5 micron, and PM0.1 is, you know, 100 nanometers or smaller. So we're seeing that the stuff that is sub-micron, you know, and the 2.5 micron and smaller represents about half the total particulate matter when we in our samples, which one of the benefits of using a time of flight instrument, and you know, it's maybe a bit overkill, but we are able to collect a lot of data on other things that have been introduced to the instrument. So for example, you know, at the end of a hard day, you go home. What do? You sit down on your couch, and whoosh, you know, as you push down on the cushion, dust is going to be emitted to the air, and it's probably mostly polyurethane foam particles that make up the foam padding. So we asked the question, well, do we see other polymers in our samples? And in this particular case, identify polyurethane foam based on its degradation product shown here. And I'm showing the retention time, and sorry, this is the drift time, the retention time, and the mass spectra obtained from the sample compared with a standard. You know, it's very easy to go grab a little bit of polyurethane off the shelf and inject it into an instrument. We're seeing the same signature. So we're fairly confident we're able to identify that. We're also able to identify small molecule contaminants that could potentially be riding along for the ride with these particles. So for example, TDCPP, this chloroorganophosphate ester, is a well-known flame retardant that is often used in polyurethane foam, whether you know, as a cushion or a foam, or sometimes polyurethane is used as a sealant. We see that there's a relatively good correlation between the measured concentration of the polyurethane foam and the TDCPP signal observed. So what we're hoping to get at eventually is to identify the polymers, but also the small molecule pollutants that are following along. And I mean, why are we concerned about this? Well, this is a compound that has been studied before and that has been shown to be problematic in the environment, and potentially will also have adverse effects. At the end of the day, and I'm just hypothesizing, maybe it's not the plastics that we have to worry about. Maybe it's the other thing, the additives to the plastics that are more problematic. So now I'm going to switch gears a little bit and talk a little bit about what Michaela Rousel did. Beyond the first two samples, she managed to collect air samples from 10 different locations in St. John's, and we managed to get data from about 8 of them. And this shows a breakdown of the particulate matter 10 and 2.5 compositions. She did a gravimetric analysis, so basically just weighed the filter and then did a mass balance with what she was able to quantify of plastic. And the conclusion was that up to 65% of the indoor particulate consists of plastic. This is a pretty high number, and it's largely driven by one sample. So I admit that right away, but it does give an indication of that plastic is maybe a significant part of particulate matter, if not the dominant part. We found that in this preliminary study that most of the plastic detected is polystyrene, and that's really strange. I mean, it could be that because polystyrene is not very dense, certainly expanded polystyrene foam is not very dense. It could be that that's floating around as more available to the air sampler than other plastics that maybe. That's possible, but I mean, it's early work yet. I'd like to collect some more data to see if that's really the case. And I mean, this is just a quick look at the concentration of the plastics that we observed with respect to the age of the home. And this was rather surprising that there's a loose trend of increasing plastic concentration with decreasing age. That's actually kind of surprising because other pollutants that have been measured, like brominated flame retardants and plasticizers that have been measured in dust, have shown opposite trend of tending to be higher in older, dustier homes. But again, this is an N equals 8 experiment, so I think we need more data to dig into this.
And like this is because more and more things are made out of plastic in your house, like even flooring now is like a vinyl or like is has some plastic layers to make it maybe waterproof. That's why, or like maybe walls, or I don't know, like think of more and more plastic.
That's certainly possible. It could be reflective of the trend in construction materials. We don't know. Maybe it's just because it's in St. John's. I would have to try this in a few other locations. But it's interesting to look at as at first glance. Now, an often quoted statistic, which I totally disagree with, is that we eat credit cards worth of plastic every 2 weeks. That comes from not WHO, but a WWF report out a few years ago that didn't seem to have any hard literature references associated with it, any like peer-reviewed science. So I have some difficulty believing that, and other people I think have the same skepticism. So there's this paper by Martin Pletz that reviews this question and the available information literature and comes up with a much lower exposure level. So the question I had was, okay, well, based on what we've measured, how do our concentrations or measured concentrations compare to that crazy statistic? And we conclude that we're inhaling up to about 200 micrograms of plastic per day. And admittedly, that's quite a bit higher than what's been more recently proposed as the daily intake rate. And who knows, we might be off with our values. It's a difficult analytical problem, and I think we should allow for some uncertainty there. But it's certainly less than a credit card a day. So a credit card, if it's 5 grams, it would still assuming it was made of polystyrene, not PVC, which that's what they're made of, it still takes something like 85 years at the rate we've proposed to inhale. So you're safe. You're probably not consuming that much plastic. We don't necessarily know what a safe level is. Why our group became concerned in the first place was this study here that as full disclosure, I was involved in. If you feed mice micro and nanoplastics at elevated concentrations, polystyrene nanoplastics will cause fetal growth restriction. So what this data here is showing is that the fetal weight, the weight of the babies, decreases with increasing concentration of exposed plastic. So that's obviously concerning, but we really need to know what our exposure level is, what the dose is, to determine whether this is actually, whether plastics are actually toxic. So from this, we estimate that humans are exposed to maybe about 3 micrograms per kilogram by inhalation alone. And the adverse effects start around 5, but really it's, you know, an order or 2 magnitude of seeing effects. So I would say that, you know, the levels that we're seeing are actually reassuring that maybe we shouldn't be alarmed about these exposures, but still work needs to be done to refine the analytical work and to get a more comprehensive picture of what our exposure is. So the conclusions are as follows. I've shown you a method to identify and quantify micro and nanoplastics indoors. We estimate that we could be breathing up to 200 micrograms of plastic per day, and that the concentrations may be somewhat higher in quote newer homes. Up to 65% of indoor dust could be plastic, although that was only the case in a couple of samples. And we estimate that maybe it probably takes 80 to 100 years to inhale an entire credit card's worth of plastic. So the future work we're aiming to do focuses more on the small molecule additives that are present in the plastics. And it's possible that these, and it's, others have suggested it, that these plastics serve as vehicles for exposure to other compounds like flame retardants and plasticizers that are maybe more harmful than the plastics themselves. So we want to characterize those as well as the other polymers present in the sample. And finally, I just want to thank the people that made this possible. So I don't get to do any work anymore, right? It's the students, especially Maheen here, who was the lead author on the paper, and Michaela, who did the work during the summer, expanding the research to more locations. So thanks to them, and thank you all for your attention.
Thank you very much for the presentation. I do have a quick question about the ion separation itself. Like, so you're using this cyclic ion system. You said like you're doing only one pass through the IM cell, just one turn. Is that because it would take more turns, like versus the pi GC timescale and the QTOF timescale, just because one pass is enough to have decent separation of the PS, PMMA, and the PP and the other polymers?
So increasing the number of passes will increase the quality of the separation, without a doubt. On the time frame of a gas chromatography experiment, where your gas chromatographic peaks might be 5 to 10 seconds wide, we estimate that you could send the ions around that cyclic cell up to 50 times before you start losing. What can happen is your GC peaks start to coalesce, and that's an artifact of those higher pass experiments. We didn't do that for the first time around for a couple of reasons. One is these are experiments that require the best sensitivity, and although you don't lose that much by doing multi-pass experiments, I'd rather have more signal and less separation for these types of experiments. And the other issue that we have to contend with is wraparound. So when you're doing these full scan experiments, the lighter mass ions will essentially catch up to the slower ions, and that can convolute the spectrum a little bit. It's something that we can deal with post-processing, but as a first pass, we prefer to just do single pass experiments.
So we have 4 raised hands. So I think Chris, we were first.
Yeah, I have 2 comments, but I'll ask one question, and then I'll circle back around, I guess, is how we'll do that. My first one is, I thought that was very fascinating work, and I want to just point out that and this is more of a comment than a question, you were probably very wise to point out the difficulty of quantification by pyrolysis GCMS. It's a very common question we get asked within industry for, and it's incredibly challenging and basically nearly impossible at times. But I think so. If you can go back to that slide, I guess my question would be, just the process of your quantification. You're doing that based off of mass balance, essentially, right? The total response, based off of concentration instead of one particular analyte, or how? What is the actual one more slide back, I guess, on how you set up the calibration curves. That would be based off of, yeah, just total response, or on the chromatogram, or one particular analyte in that.
Right. So one thing I didn't talk about that actually might have been useful for an ASMS group, apologies, are the specific target ions that we're monitoring. So most of the pyrolysis GCMS work that I've seen so far focus on detecting by selected ion chromatogram relatively low pyrolysis products. So for example, polystyrene produces a monomer, dimer, and trimer pretty readily at 500 degrees or so under our experiments. And we monitor the trimer, which is mass 312. And that's actually what most people would do, even if you had a regular old EI single quad mass spec. For the polypropylene and polyethylene samples, on the other hand, what I've seen in other work is that the products are not particularly, like, they're alkene products. They're not very specific. They're not. And there could be lots of other things that produce those small pyrolysis products. So one advantage of our setup is that we've coupled GC to the instrument by APCI. I mean, that's nothing special. It's what Waters offers to connect their GC, and that allows us to monitor much higher molecular weight pyrolysis. We're monitoring, I'm trying to think. Actually, let me look. Yeah, we're monitoring like C43 and C34 for the polypropylene and polyethylene polymers, respectively. So it's less likely that there are other things present in your sample that can break down. And I think that's going to be more important as we tackle biological samples, where lipids and things like that can potentially degrade into the same products. But to answer your question, yeah, it's selected ion chromatograms of a few of the pyrolysis decomposition products, and then we're doing external calibration. We have purchased isotopically labeled polymers, but we haven't actually done anything with them.
Yeah, and what you'll find also is that if you're doing a normal pyrolysis procedure without the APCI but using EI instead, you'll actually get the oligomers start to creep up on you, because it's a reactive process, pyrolysis. And so if your concentration's increasing, you'll start to actually see more and more of the Z-mers and oligomers retain in your chromatogram and stuff like that, and it becomes quite challenging. And so I was very, very curious at how much of this is accounting for those sort of concentration effects on the reaction that are occurring. So that might be why you're getting those large distributions as well, that you might not be accounting for even larger things that aren't ionizing by APCI even. But anyways, it was fascinating work.
Thank you.
Thank you. Hey, we have Scott next. Yes.
Yeah. So I have a question. It seems like you have a lot of polystyrene. Is it that because the polystyrene you get more interaction, or is you just have that much more polystyrene than polyethylene, polypropylene, and PMMA?
That's a good question, and it's something that we're thinking about. So, this is the first attempt at it. Our original explanation for this is, well, maybe expanded polystyrene foam is more available to the air sampler because it's lighter, less dense, it's floating around. But I've seen, I've also seen that the response factors, and this is, work that's been communicated to me by others. I don't want to, say who or what, but depending on the polystyrene source that are standard that you use, the response factors can be quite different. And I mean, I'm looking to the polymer community to maybe comment on that. because I think that could be a significant problem if we're trying to do this. Now, as a first shot at it, are we happy with an order of magnitude estimate? As an analytical chemist, it's always good to do better, but it's at least we can say we can provide a rough estimate of the exposure and go from there.
Okay, great, thank you.
Thank you.
Chris, you had a question too?
Yes. Yeah, I pretty much mine is related to what Scott asked. You would expect in an indoor room more polyethylene based on the plastic bags that we use, not polystyrene. But anyway, when you pyrolyze those polymers, polystyrene has those benzylic bonds that will break up much more easily. So I don't know, do you take into account how easily those things pyrolyze into products and that you can see in the GCMS? So you can obstruct the concentration profiles through the pyrolysis process if the one polymer like polystyrene pyrolyzes more easily.
So, I mean,
polystyrene polybutadiene I would expect to pyrolyze, to break bonds more easily because they are allylic bonds and they are lower in strength than the polyethylene. The CC bond is the strongest.
Right. So, I mean, we would hope that would be captured by preparing the external calibration. I agree with you that polyethylene is by far the most abundant and mass-produced polymer. So one would expect to see that. Certainly in the marine environment, it floats and it tends to dominate those analyses. So I'm not sure how to answer your question other than we're not sure either why we're seeing this.
I will interject and say that when you count for indoor surfaces, the surface area of a room is going to be consistently paint for accounting for a lot of that, not just bulk materials, and that's going to be PMMA and styrene. So that could be an accounting for that.
It could be. Now, when you see this slide that you show here, the y-axis polystyrene has a very low response factor compared say, to PMMA, polypropylene, and polyethylene. The fact that you see lots of polystyrene, that enhances that. You see the response?
That's correct. And so you have to assume that these response factors are right, and maybe they're not. This is our first attempt at it, and I've communicated with a couple other groups on this, and one of the pictures that seems to be forming is that depending on the type of polystyrene, whether it's cross-linked or what molecular weight, it could have different response factors.
I would recommend that you take a mixture of polystyrene and polyethylene, a 1-to-1 mixture, either molar or weight to weight, and you try that as a caliber. You try to see what do you see when you take a mixture. You may be surprised if you only see polystyrene, or that will give you some explanation of all those questions that we raised now. It's very interesting, very exciting, as previously said. And so this type of experiments probably will reveal more light on that if you analyze the mixture. Like things that were done to figure out the moldy efficiency. You remember Kevin Owens and other scientists took mixtures to analyze them to see what's the difference in the efficiencies. And you can do the same thing now with your method to see if you have a 1-to-1 mixture of whether one is advantaged versus the other.
So this data that's shown here, I'm quite sure was collected by mixing the polymers together. So it's
I suspected it's individually, every polymer individually.
No, they were mixed.
They were mixed?
They were mixed. But I mean, maybe one polymer is suppressing another. I mean, it's a fairly complex process that's happening in the pyrolyzer that sometimes us analytical chemists don't think about. At least I'm not thinking about.
Yeah. A binary mixture would be easier to interpret then. Yes, that's right.
Yeah.
I think Luke also had a question.
Yeah, I was I guess we're all going on the concentration question here and the calibration. So I think I emailed you about this before, but I was curious, what was the format of the samples being quantified versus the format of the calibrants? You talked about, about suspensions, and I was curious if the calibrants used were like water suspensions, or, you mentioned earlier about doing a hot toluene extraction, and then later you found out you could do a methanol DCM suspension. And my mind just keeps going to, these are dissolutions, these are all extractions and dissolutions and mixes thereof. And I'm curious how exactly that the format of what's going into the pyrolysis tube is the same format, both particulate, and extract, as the calibrants. So maybe you could walk us through the format that is the solvent mixture and how the sample was treated versus the solvent mixture and how the calibrants were treated.
Okay, so the way that these standards were prepared essentially involved dilution in heated toluene. And as you mentioned, what's been done in other groups is you might take a solid aliquot of the plastic and use accelerated solvent extraction to make a solution. We haven't done that here. We found that it's more convenient to do the dissolution in a mixture of methanol DCM that roughly matches the density of the polymer. And I don't know what the best way is. I suppose one could evaluate that by determining which results in the highest response. As far as the samples go, they were, your polymer is embedded in the glass fiber filter. We put it in an accelerated solvent extraction cell, extract it, take the solution, concentrate it to about 200 microliters, and then we transfer about 10 microliters to the pyrolysis cup. So they're not being traded exactly the same, and that could be, where some of this could be an issue. We just have to try. Does that answer your question? I'm not sure that I answered your question, but I'm not entirely sure I caught it.
Well, I wanted you to walk us through it, and you did, and so that answers a lot of questions. I mean,
okay, good. Okay,
yeah,
good.
There's still
sure.
the judgment of, in what form is that, and how does that affect the pyrolysis? But now it's clear, and I don't think I necessarily have the data that tells me, oh yeah, in that format it will do this, But at least now we know the formats and any differences.
So what I take away from this first, shot at things is, yes, it's a difficult experiment, but even if polystyrene's response factor is low, we're generating rather conservative numbers if we're trying to assess exposure. Low response equals high concentration, and those concentrations we're observing are still lower than we would expect to cause an observable effect. So I'm convinced that, as we gain more experience with the instrument and with the method, we'll get better quality numbers. My main concern right now is to make sure we don't overstate anything by saying something silly like, oh, there's plastic everywhere, and and this is really harmful. We don't actually know that, and I think I've seen that. That is something that I've seen in other studies.
I have a very quick question about the APCI, because I'm not familiar with this GC APCI versus GCCI. How does it compare? Like if you overlay a spectrum like GC, like a PyGC-CI versus PyGC APCI, it looks the same, or you have a different fingerprint because of the APCI? I'm not very familiar with it.
So I get the impression that that polystyrene behaves quite similarly between EI and APCI. if you look at, there's the picture, polypropylene and polyethylene, we don't have an EI system, so I'm only going based on anecdotal evidence of talking to people. I sort of got the impression that they don't need abundant pyrolysis product has up to 30 or 40 carbon atoms. I sort of got the impression that it was the lower molecular, it was skewed more towards lower molecular weight. So that could be a result of the ionization.
And in terms of like instrumentation, like when you do APCI, there is no need for any gas, right? Like you don't add any methane or as a propane or ammonia. Just with the AP, it's enough to have ions.
So we're working under charge exchange with charge exchange conditions under nitrogen.
Okay.
So N2 and N4 plus. Oh, there's a question from Chris.
Yeah, I think, yeah.
Actually, thank you for showing the spectra again. That could explain the nice response for polystyrene. It's essentially monodispersed the pyrolysis product, just those two products. So all polystyrene goes to the dimer and trimer. You get the distribution with the other ones. It's like having a monodispersed polymer versus a high polydispersity. So that could have also with the detection efficiency. See, polypropylene and polyethylene, you spread it over many oligomers, and polystyrene is the nicest one that you get only 2 out of that.
That's a good point.
That's very nice. I mean, you showed that, but I paid attention now that you showed it again.
There are mathematical ways of treating that I think theory can probably talk to you more about with KMD at some point. But yeah, that is a nice point, Chris.